Hello everyone, my name is Bhargavi. I'm a PhD scholar at Amrita School of Biotechnology. My presentation for today is titled Integrated System Genomics Approaches for Deciphering Candidate Genes that are Regulating Prostate Cancer Phenotype in India. A prostate cancer is a adenocarcinoma that develops in the glandular part of the prostate gland. The major risk factors for developing prostate cancer are age, ethnicity, obesity, family history, lack of exercise, persistent elevated testosterone levels, etc. I have chosen prostate cancer for my research because there is a serious gap in the knowledge base of candidate genes that are involved in prostate cancer phenotype, particularly in the Indian population. There also seems to be a missing link between diabetes and prostate cancer. Prostate cancer is a fairly common cancer, being the second most prevalent cancer worldwide and the third most prevalent cancer in India. Um, as part of the CAPSI consortium of India, which is the only consortium that deals itself with whole exome sequencing of prostate cancer samples um, and performing genomic analysis. A pilot study was conducted by Gupta and Shukla et al. In 2020, where they uncovered mutations in genes that are associated with obesity, diet, mitochondrial dysfunction, and vitamin D deficiency in a prostate cancer cohort from India. The mutations identified are represented in the picture on the right. The objectives of my research include the identification of lead mutations that are influencing diabetes in a prostate cancer cohort from India and performing whole exome sequencing to identify the variants and also find possible links to diet and lifestyle. Um, I also aim to identify candidate differently expressed genes including long non-coding RNAs and checking their regulatory nature using a fly model. Pictured here is the benchmark pipeline for our whole exome sequencing, and the variants identified through this will then be validated using a fly model where we're using axillary gland and drosophila as a model to study prostate cancer. Axillary gland and drosophila is a functional equivalent of the prostate gland, hence this will be used for the transcriptomic analysis. A cell type specific transcriptomic analysis can be achieved without RNA isolation using the targeted DAM-ID method, where we will be isolating methylated DNA for Pol II occupancy, and the gene expression profile obtained uh, uh, will be able to provide us information of differentially expressed genes. So the comparison of the transcriptome of wild type axillary gland and the proliferated axillary gland uh, will shed some light on the overexpressed or underexpressed genes in both cases, and this will then be screened through our candidate genes that have been identified through whole exome sequencing in the prostate cancer cohort in humans. Through this work, we are hoping to identify risk SNPs that are associated to prostate cancer in India, identify a possible association of diabetes with prostate cancer, and also identify cancer genes that are expressing both in the prostate gland as well as in the axillary gland, and also perform functional analysis of those genes to identify protein products or RNA products. These are some of the references. Thank you.